Welcome to my YouTube channel, Christy's Crash Course. Please check out the previous video as this video is only a continuation on the subject matter thyroid hormones and its synthesis. Stick around to know more. The pituitary gland releases the TSH. Remember that the TSH means the thyroid stimulating hormone. The TSH, so this is it. The TSH goes to bind to your thyroid stimulating hormone receptor, which is found on the cell membrane, right? On the cell membrane of the follicular cell. So this is our follicular cell. Follicular cell. Remember that the thyroid gland has two major cells. It has the follicular cells and it also has the calcitonin or C cells. And from this diagram here, right, you could easily see how that one follicular cell is joined to the next one, which is joined to the next and to the next and to the next and to the next. So we are looking microscopically on just one or two follicular cells precisely. And because of that, we should understand that surrounding each of these cells, we will naturally find blood vessels, right? Because cells needs the blood or the plasma for nutrients. So naturally we'll find blood vessels here. Blood vessels. And why is this blood vessel important? Because within the blood vessel is how the thyroid stimulating hormone also gets to the thyroid stimulating hormone receptor found on the cell membrane. Now, when it gets to this receptor, we explained in the previous slide that we get to see the activation of G stimulatory protein and GQ. So here, G stimulatory and GQ proteins, and the G stimulatory protein activates the adenine cyclase, which eventually activates the CMP and goes to activate your protein kinase A. Now, protein kinase A has the ability to activate a variety of enzymes which work to eventually cause for the production of the thyroglobulin. Remember, we said that thyroglobulin is so important because it is the platform on which our thyroxine hormones can be formed. Because of that, thyroglobulin must need to be activated. It must need to be stimulated so that there could be a transcription process, a translation process, and eventually the rough endoplasmic reticulum can produce proteins, and then we can have the Golgi apparatus work on it and give us a fine thyroglobulin. So these are multi-steps that brings us to our thyroglobulin molecule. Okay, now if you follow through the cascade, you will see that thyroglobulin will eventually leave the follicular cell and go into the colloid. So we have to assume that our white background here is the colloid. So the colloid we are seeing here is what is inside here, the colloid. Okay, so what happens when it leaves into the colloid? So we're going to imagine that our thyroglobulin has come out and it's just somewhere here in the colloid. So we'll leave that there. So the next thing we have to look at is how does iodine comes from the blood into the follicular cells. Now it's important we know that iodine is gotten through our diet, what we eat, and so in the blood it is in a form known as iodide. Okay, iodide. Now for iodide to come into your cell, it can't do it by itself. It actually needs help. And it's very interesting because when we get to talk about the membrane potential in a cell, whereby we get to see that within a cell it is naturally negatively charged and outside the cell it's naturally positively charged in reference to the inside of a cell because of this sodium potassium ATPase. Now sodium potassium ATPase is a very very important and intrinsic pump, because it helps to pump out 3 sodium out of the cell and pumps in 2 potassium out of the into the cell. Because of this, the number of sodium being pumped out. So if you look at the ions, you could see that sodium is positively charged, potassium is also positively charged, but in the long run, the amount of positively charged ion pumped out of the cell is more than what is pumped in. So eventually, the cell will have a negative membrane potential. Now, it also tells us that the concentration of sodium outside of the cell is higher than what will be inside the follicular cell, and because the principle of diffusion works, sodium will naturally want to flow into the follicular cell, and so sodium goes in based on the electrochemical gradient already created due to the electrogenic effect. So we call it the electrogenic effect of sodium potassium ATPase. And so due to this electrogenic effect, we get to see that sodium comes out. Sodium can eventually go into the cell, and because of how much the gradient it has, iodine also can be tagged along and can enter into the follicular cell. So that's the advantage that these secondary active transports have. They do not need ATP like the sodium potassium ATPases. They don't really need that. Rather, they just make use of the electrochemical gradient that is already created due to the massive amount of sodium outside the cell. So sodium is very high outside and very low inside, and so it can easily just carry along iodide. Now, it is important to know that when iodide entered into the follicular cells, its interest is not really to stay in. It actually wants to go out. It wants to help in the process of the formation of thyroxine, thyroid hormone eventually. And so what happens? We have a very, very, very unique channel, and the name of the channel is called pendrine, right? We call it pendrine, or we call it chloride iodide exchanger. Okay, chloride iodide exchanger. And why is it important? It's important because it brings iodide into the colloid. Remember the colloid, while it can also take in chloride into the follicular cell. So exchanger means one is going in a particular way, another is going in an inverse way, and so that is what this pendrine does. This pendrine channel is highly important because in a case of mutation of this channel, we can actually find that such persons will be more prone to having goiter and hearing loss, and so pendrine channel is very important. So when iodide is brought into the colloid, we have a strategic conversion of iodide into iodine because we want to iodinate these tyrosine residues found on thyroglobulin. So iodide with the negative, so the iodide with negative, we want to convert it to iodine. And this process of conversion from iodide to iodine requires a peroxide, so to speak. And so the process of conversion, we need our dual oxidase 2. Now you could see these are very, 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 very strategic pumps or strategic enzymes that are located on the apical membrane. It is important that we remember that there is an apical and there is a basal membrane. The apical membrane faces towards the colloid, the basal membrane faces towards the blood vessel. Okay, and so within the apical membrane, we have our dual oxidase 2. Our dual oxidase 2 uses oxygen and forms hydrogen peroxide. Okay, and then when hydrogen peroxide is being formed, our thyroglobulin peroxidase uses it to convert our iodine into iodide into iodine, and so when it is being converted, then your thyroid peroxidase now takes the same iodine and attaches it to your thyroglobulin. Okay, it attaches it to precisely the tyrosine residues found on the thyroglobulin. Remember our previous slide, we said that if thyroglobulin is something like this, then we have tyrosine residues anchored on it. So the goal of this process is that we want to anchor our iodine onto these tyrosine residues. And if we anchor it in 2 points, so if we anchor it at the 3 or the 5th position, we could form a diiodothyronine. If we anchor it only at the 3rd position, we could form a monoiodothyronine. So first things we have to consider is that there has to be an oxidation process. An oxidation means that we are converting the iodide into iodine, right? And for that to take place, we needed the hydrogen peroxide. And then just after the oxidation process takes place, the next thing we are doing is that we want to ensure that this iodine is being bound to our tyrosine residues on our thyroglobulin, and this process is called the iodination process. And eventually, when we're able to form the monoiodothyronine or the diiodothyronine, we want to couple it together. Why? Because we said that DIT plus DIT will give us a T4. Okay, and we also said that MIT plus DIT will give us a T3, and these are the 2 major active forms of our hormones. 2 major active forms, T3 and T4, and that is actually the end result we want to get. And so eventually you get to see that when we get to have this thyroglobulin that has been iodinated, and then we want to use it because we don't want it still bound to the thyroglobulin molecules. We've been able to successfully get T3, T4, but we want them as independent molecules. So what do? We can endocytose it, and then when it's endocytosed back into the follicular cell, then we have a conversion of or a joining together with lysosome. So these cytosed thyroglobulin molecules is being phagocytosed by the lysosomes, and then we have proteases acting on it so that it can cleave off T3, T4, cleave off MIT, DIT, so that they stand as individual substrates. And eventually we get to push them back into the circulation. So assuming this is our blood vessels, right? It's pushed back into the circulation. Something else we need to know is that when it's pushed back into the circulation, they are actually binding globulins, which are normally produced by the livers, that helps to hold these hormones, because they are not able to freely flow in the blood like maybe other peptide hormones. So what happens is that we have thyroglobulin binding globin, we have tetraetrine, and we have albumin. These are structurally important binding globulins that anchors to the T3 or the T4 of thyroid hormones. Let us then attempt some questions about the subject matter that we have discussed. So the first question says, which transporter is involved in transporting iodide out of the follicular cell at the follicular lumen? So remember that when we are considering the subject matter of iodide, when we look at iodide, we must remember that it is still in the negative form. So iodide is this form, and so word transporter moves iodide from the follicular cell into the lumen. Let us go and check from our diagram. Perfect. So if you can see, when we considered the iodide, we said iodide was inside the follicular cell here, right? So iodide came in, and then it moved through PDS, or we called it the pendrine, also called the chloride iodide exchanger, and it was being pumped out into the colloid at the follicular lumen. And so, that being said, word transporter, we call it the pendrine transporter or the chloride iodide exchanger. So the first one says sodium iodide sympather. No. Why? Because the sodium iodide sympather, NIS, is used to pump iodide into the follicular cell, not out, right? Are you seeing it? Into, keyword is into. Then the next one, potassium iodide sympather, it's not a transporter even in the context of thyroid synthesis. And then we have pendrine. Pendrine is actually the answer in this case. Pendrine is the answer. The next question says, what is the function of thyroglobulin in thyroid hormone synthesis? This is a very exceptional and beautiful question, and it says A, transporting iodide into follicular cell. So this cannot be the answer because we just saw that what transports iodide into the follicular cell is the sodium iodide co-transporter, actually, because it works on the principle of the electrochemical gradient. Then we have acting as a substrate for iodination and hormone synthesis. Can that be correct? Regulating sodium iodide sympather, not really. It doesn't regulate. More so, what really wants to regulate the sodium iodide sympather is the, we call it NIS, is usually our TSH, okay? But then for converting T4 to T3, now this is a very, very important part of our discussion. And why is it important? Because there are specific enzymes, so enzymes that convert T4 to T3, or even converts T4 T3 to DIT and MIT, or convert T4 to reverse T3. They are enzymes. So the name of these enzymes are collectively called deiodinases. The iodinases, they convert, and that's why even though we get to have a high amount of T4 being pumped out into the circulation, it is even said that it is in a ratio of 10:1. Even though we have that ratio of T4 to T3, you could actually still see that when this T4 goes into the cell, the receptors that are receiving the thyroid hormone are more sensitive to T3, and so eventually in extrathyroidal tissues, T4 is being converted to T3 for the cells to use it, and also for other cells to, take it in and rot the function of metabolism and maintaining the basic metabolic rate. Now it leaves us with option B. So option B says acting as a substrate for iodination and also for hormone synthesis. Is that true? Well, let us go back to our diagram. if we come back to our diagram, we for sure saw that thyroglobulin came out and it was a substrate all along. So it came out, it had to join with iodine, right? And then it had to couple with there had to be a coupling of the tyrosine residue to form our final product, which was later broken down by the proteases within the lysosomes in order to give individual hormones here. So eventually option B is the correct answer. We're going to do a summary of everything we have looked at in this video and in the previous video. We are going to start with the very first process of thyroid hormone synthesis, and so you could follow along with me, right? So while we bring up the processes, you could also try to pause between the videos and try to see if you can remember something, if you can save exactly, get the channels or the transporters or the enzymes correctly. Alright, so the first process is good. The hypothalamus releases the thyrotropin releasing hormone from the paraventricular nucleus. Paraventricular nucleus is important here. The second one is the thyrotropin releasing hormone bind to the thyrotropin releasing hormone receptor on the thyrotropes of the anterior pituitary gland, because thyrotropes will produce the thyroid stimulating hormone into the circulation. Great. The next one is TSH binds to thyroid stimulating hormone receptors and activate a cascade of events via the GS and GQ pathway, which eventually ends with thyroglobulin synthesis, sodium iodide synthetase expression, and thyroid peroxidase expression. Of course, when we looked at each of these pathways, they can branch very much into other things. Alright, going on, the thyroglobulin is exocytosed into the colloid. Then the iodide is transported into the follicular cell along sodium via the sodium iodide synthetase. Next, iodide is transported out of the follicular cells via the pendrine or chloride iodide exchanger. And we have the iodide is converted into iodine, which is a more neutral form, by thyroid peroxidase, or we call it TPO, using the hydrogen peroxide. And usually the hydrogen peroxide is formed from dual oxidase 2 enzyme, which converts oxygen to hydrogen peroxide. Alright, the iodide is added to the tyrosine residue on the thyroglobulin molecules in a process called iodination. Good. Then we have DITs, which is diiodotyrosine and monoiodotyrosine, conjugates to form T3 and T4 with the help of TPO, which is thyroid peroxide. Then we also have the hormone alongside thyroglobulin molecules are endocytosed, so they are taken into the follicular cells. Next one, the endosomes fuse with the lysosomes to form an endolysosome. Proteases, which are mostly the lysosomal enzymes, acts on the thyroglobulin molecules and gives rise to T3, T4, DIT, and MIT. We finally have our active form of thyroid hormones. T3 and T4 are exocytosed from the follicular cell into the circulation. 3 important binding proteins we need to remember is the thyroid binding globulin, the transthyretin, and the albumin. These three proteins are important, and they have much more affinity to bind to T4, somewhere around 99.98%, while for T3 they bind less, that's 99.5% is being bound to T3, which is a thyroid hormone, the triiodothyronine. So this is the summary of the thyroid hormone synthesis. I really hope it made sense to you all. I really hope you understood it. Thank you so much for staying till the end. Do well to like the video, subscribe, and leave a comment, leave your questions, leave your reservations about the video in the comment section, and I will see you next time. Bye.